Now this is the recall session of FMGE exam held in January 2023. So we will be collecting all the questions and we'll be discussing them one by one, right? So, all right, so let's start with the discussion. There are so many questions and the paper was very balanced paper. They had a lot of clinical questions as well as recall based question as well as application based question. So let's start discussing biochemistry question one by one, and all of you are free to interact and discuss, right? If you recall any other question, you can always discuss that as well. So now the first question which I am projecting here is completely recall based, and here they said that there is a patient who is presenting with liver damage and examination of eye is showing the following finding. This was a picture which was given, and now which of the following factor may be responsible for such presentation? So in this question what they have given is just the liver damage and the eye finding, where you can see there is a sort of opacity in the lens, and if you see the appearance, it is a sort of sunflower appearance. So it is a sunflower cataract. So this is a very characteristic finding of Wilson's disease, where due to excessive deposition of copper in liver and various other organ system, there is a damage of those particular cells. So the answer for this question is the copper, because in Wilson's disease, there is a problem in copper excretion from the body. So what is Wilson's disease? Wilson's disease. You remember one protein, that is copper binding ATPase 7B. This is copper binding protein which is helping in copper excretion from the hepatocyte. This is mutated in Wilson's disease. There is something called copper binding ATPase 7A as well, but that is seen to be mutated in minky kinky steely hair syndrome. So there are two things which you must know which are due to defect in copper binding transporter protein. One is Wilson's disease, another is minky kinky steely hair syndrome. In this, the similar protein is defective, but that is a GIT mucosal protein, and that name is copper binding ATPase 7E. So this is 7A and this is 7B. This Wilson's disease protein, copper binding ATPase 7B, is present in hepatocyte, which is responsible for excretion of copper from the liver to the biliary canaliculus. From there they are getting excreted out. So excess copper is getting excreted out. So when this protein is mutated, then what is going to happen? There will be a lot of copper deposition, in the hepatocyte, excessive copper deposition in the hepatocyte. Liver is not able to get rid of this copper. So what happens? This copper toxicity results and liver is going to damage. That liver will undergo cirrhosis. So that is Wilson's disease. And this copper binding ATPase 7B is not only responsible for excretion of excess copper, it is also responsible for secretion of copper from the liver, and so that's going to bind with something called as apoceruloplasmin. Apoceruloplasmin is a protein which is going to bind with the secreted copper, and that's going to make you ceruloplasmin. So in Wilson's disease, this is characterized by low ceruloplasmin. See, apoceruloplasmin level is normal, but because copper is not binding with apoceruloplasmin, the ceruloplasmin concentration is going to be less. So in plasma, when you see, you have low level of ceruloplasmin, and liver is having excessive copper deposition. So when you do the liver biopsy, you have got a lot of copper there. So that's a characteristic finding in Wilson's disease. Now there are many organs which are affected, like for example I was talking about the liver. Copper toxicity damaged the hepatic cell, resulting in cirrhosis, liver failure, and then copper also gets deposited in undergo deposition in brain, resulting in psychiatric features, right? Psychiatric manifestation. So copper deposition in the brain results in psychiatric manifestation. Copper also deposits in eye, and there you find the damage of the lens that is giving you sunflower cataract. Sunflower cataract, which was shown in this question. And you must know, when we talk about the cataract, there are appearance wise you have so many types of cataract. As you can see, you have oil drop cataract. Can you appreciate this oil drop cataract? This is seen in galactosemia. Galactitol is the factor which is responsible. Galactitol. Galactose is reduced to galactitol by aldose reductase, that is causing this oil drop cataract. You can see, appreciate this oil drop cataract. When the cataract is snowflakes in appearance, you can see snowy cataract, that is seen in diabetes, which is due to deposition of a lot of sorbitol, which is hygroscopic, and this is reduced form of glucose. It's an alcohol of glucose. This also is produced. Sorbitol is also produced by aldose reductase enzyme in the lens cell. This is the brown ring which is seen around the cornea. This is called Kayser-Fleischer ring, and this is a characteristic feature seen in Wilson's disease. Wilson's disease, in addition to sunflower cataract, you have this kind of brown ring around the cornea seen in Wilson's disease, and this is called Kayser-Fleischer ring, and this is due to deposition of copper in descent membrane of the cornea. Descent membrane of the cornea. So this is copper deposition in the eye, which is resulting in the cornea. It is causing you KF ring, which can be seen by the naked eye, as well as you can examine in slit lamp. If it is in milder form, you can examine it in slit lamp to find this KF ring. This is called as golden brown. You must remember the name also. Golden brown color ring. It's a very characteristic feature of Wilson's disease, but not seen in all cases. Less than 10% of the cases are going to show you KF ring or case of, right? So this you must know. So this question was very clear-cut question, because they talked to you regarding the liver damage and along with the sunflower cataract, and it is a Wilson's disease which is due to copper toxicity. Glucose toxicity seen in diabetes in hyperglycemia results in snowflake cataract. Galactosemia results in oil drop cataract. Mannose, there is no condition where mannose will be high in the plasma, and that is not going to cause you any sort of cataract. All right, any doubt in this? All right, now coming to the second question. Second question was talking about a graph, where a graph was given, and the graph was like this. If you see, there were two lines which were diverging, and they asked that which type of inhibition it is. So the answer for this question was non-competitive inhibition. This is double reciprocal plot, or otherwise called Lineweaver-Burke plot. It's called double reciprocal plot. Double reciprocal plot. Reciprocal plot, or otherwise called Lineweaver-Burke plot. Lineweaver-Burke plot. B-U-R-K. Burke plot. This is the double reciprocal plot which talks about enzyme kinetic in presence of and in absence of inhibitors. So let's talk about this, how I said that it is a non-competitive inhibitor. Line. See, in Lineweaver-Burke plot, you have a very characteristic finding. Based on the type of inhibitor, you have a very characteristic picture on the graph. First of all, you must know what is Lineweaver-Burke plot. It's a double reciprocal. It's a double reciprocal plot where one by velocity and one by substrate concentration is plotted respectively on y-axis and x-axis, right? This is 0 point, and this is the straight line which is representing you enzyme kinetic. Enzyme, pure presence of enzyme. You get to have a straight line which is touching the y-axis at this point, and this point is denoting you the value of 1 by Vmax. First of all, you must understand how the Lineweaver-Burke plot is plotted for a particular enzyme in absence of any inhibitor. And virtually when you merge this line, retrospectively on the minus x-axis. This point represents you 1 by km value. Numerical value of 1 by km will be represented by this point where this line is virtually merging on the minus x-axis. Alright, so this is the picture of Lineweaver-Burke plot. Now when enzymes are acting and inhibitors are acting on the enzyme, that inhibitor line, if it is shown to be like this, this is seen to be a crossing line. Crossing line, you remember, C for crossing line is for competitive inhibitor. Competitive inhibitor. Competitive inhibitors. Why I said so? Because in presence of competitive inhibitor, the Vmax remains same, right? So 1 by Vmax value remains same and Km value is increased. So 1 by Km value is decreased. So this is how when you apply this concept of the alteration of Vmax and Km value in presence of competitive inhibitor, you get to have this kind of plot which is showing the crossing line. And in non-competitive inhibitor, if you see, talk about the non-competitive inhibitor, what actually happens is, whenever non-competitive inhibitor is acting, then what is the impact on Vmax? Non-competitive inhibitors, when they are acting, Vmax is going to be declined, lower value, and incidentally the Km value is going to be the same. So when this is seen in Lineweaver-Burke plot kinetic, this is enzyme, and as I have already told you, this is 1 by Km value and this is 1 by Vmax value, right? And this is 0 point. In presence of inhibitor, say for example, I just take this black color pen for the inhibitor, you have seen the Km value is same. So 1 by Km value also is going to be the same. So this point will represent you the new Km in presence of inhibitor, and because Vmax is decreased, so 1 by Vmax value will be numerically increased. So when you merge these 2 points together, you get to have a diverging line like this. So diverging line on Lineweaver-Burke plot, which was there in your question, is denoting you non-competitive inhibitor. Similar fashion, when you're talking about uncompetitive inhibitors, which are very rare in our system, uncompetitive inhibitor, the original plot will be like this without any inhibitor, which I have told you a number of times already. This is going to be 1 by Vmax value and this is going to be 1 by Km value. See, uncompetitive inhibitor is characterized by lowered velocity maximum and lowered Km value as well. In presence of inhibitor, you get to have lowered velocity maximum and lowered Km value. So when it is 1 by Vmax value will be higher value, that is in presence of inhibitor, the apparent Vmax 1 by Vmax will be higher value and apparent 1 by Km value will be also a higher value. When you merge these points together, that is going to represent you the inhibitor which is uncompetitive. This is showing you parallel line. So it is a very easy way to understand. Lineweaver-Burke plot is a boon actually to understand what type of inhibition it is. If it is a crossing line in presence of inhibitor, it is a competitive inhibitor. If it is a diverging line, it is a non-competitive inhibitor. And if it is a parallel line, it is denoting you the uncompetitive inhibition. So given question was about the non-competitive inhibitor. You can see the diverging line here. Similar fashion, any other question may be framed and asked to you. Allosteric inhibition doesn't follow the Michaelis-Menten kinetic and it shows the wavy line, sigmoidal curve it is going to show you. Not like this straight line will be shown when allosteric inhibitors are acting. Alright? Alright. Now coming to the next question. Deepu, yes, I will be teaching you there also. Right. So now coming to the next question, which is talking to you regarding a patient who is presenting to the OPD with the features of collagen vascular disorder. Which of the following deficit is most likely to be associated with defective collagen formation? So regarding this, you must know that what all trace elements are required, for collagen formation, adequate collagen formation. So answer for this question is going to be ascorbic acid and copper, because ascorbic acid and copper, they are important for collagen formation, adequate collagen formation. If you talk about collagen formation, it's a multi-step process. It's a multi-step process. The process starts within the cell, right? This is the cell and this is the extracellular space. This is the endoplasmic reticulum and these are ribosomes. This is rough endoplasmic reticulum and these are the ribosomes which are studied on the endoplasmic reticulum wall. And here occurs the synthesis of collagen. So this is polypeptide of the collagen which is produced on the rough endoplasmic reticulum entering the ER lumen, undergoing many modifications which are known as post-translational modification. There occurs the hydroxylation of proline and lysine residues. In the polypeptide chain, you have number of proline and lysine residues. To hydroxylate them, you have hydroxylase enzyme. Respective hydroxylases enzymes are there. You have prolyl hydroxylase, you have lysyl hydroxylase, and as like any other hydroxylase enzyme, these hydroxylases, prolyl hydroxylase and lysyl hydroxylase, they also require copper, they require oxygen molecule, they require vitamin C. So unless until you have adequate supply of these materials, the hydroxylation will not be taking place. The primary structure of the collagen itself will be affected. Alright, and once the procollagen is secreted into the extracellular space after multiple steps, what happens? There occurs the tropocollagen formation. This is tropocollagen, triple helical structure, and these tropocollagen are going to be arranged in a staggered arrangement, and because they have interactions between two tropocollagens in this fashion, which is called aldol condensation. So you also have the aldol condensation, which is maintaining the staggered arrangement of collagen, tropocollagens. And these aldol condensations are possible only when you have lysyl and L-lysyl residues, or hydroxylysyl and hydroxy L-lysyl residues on the opposite tropocollagen polypeptides. So this is all aldol condensation, which is requiring aldehyde form of lysine and aldehyde form of hydroxylysyl. This aldehyde form is produced by lysyl oxidase enzyme. Lysyl oxidase enzyme, which very much requires copper. So the copper and ascorbic acid, they are very, very important trace elements which are needed for collagen morphology. So three enzymes, the prolyl hydroxylase, the lysyl hydroxylase, and then lysyl oxidase in the extracellular space are required for staggered arrangement of the tropocollagen, which is going to provide this stability to the blood vessel wall and various other tissues. So either the deficiency of ascorbic acid or the copper results in collagen malformation. So in this question, if you see, so what combination you will be answering here? The ascorbic acid and copper, they are important for collagen formation. All right. Okay. Now coming to the next question. If you have any doubt, you can ask me here. This is a very much repeatedly asked question to you. Enzyme which is deficient in von Gerke disease. Von Gerke disease is type 1A glycogen storage disorder, and you must know the enzyme deficient here is glucose 6 phosphatase enzyme, right? So glucose 6 phosphatase enzyme is deficient in von Gerke disease. What is this enzyme? What it does? Where it is located? You must know. This enzyme is found in ER lumen, and this enzyme is required not only in one pathway, it is required in two pathways. Number one is the gluconeogenesis, and number two pathway is the liver glycogen breakdown, that is liver glycogenolysis. Both are source of blood glucose. So what happens when this enzyme is deficient? Blood doesn't get glucose either from gluconeogenesis or the liver glycogenolytic pathway. So baby develops a severe hypoglycemia. Severe, not only hypoglycemia, it is severe hypoglycemia, because both the sources of glucose production from the liver, that is gluconeogenesis and liver glycogenolysis, are not going to produce glucose, because this enzyme glucose 6-phosphatase is responsible for, as the name implies, it converts the glucose 6-phosphate to free glucose, right? So free glucose formation is very, very important. Without any phosphate, it should be produced so that it can cross the hepatic cell and can come to the blood. See, the phosphorylated form of glucose, or in that matter any bio compound, is not able to cross the cell wall. So for it to cross, it has to be in free form without any phosphorylation. So this enzyme role is very, very important in the process of gluconeogenesis as well as liver glycogenolysis, where terminally this is the last enzyme of these two pathways, where by so many efforts, so many steps, the glucose 6-phosphate is formed, and that is to be converted to free glucose by this enzyme, which is lumen enzyme. So when this enzyme is deficient, what happens? Of course, the free glucose is not formed, blood is not getting glucose, and baby is going to have severe hypoglycemia, right? So this is von Gerke disease, which is type 1A glycogen storage disorder. When you talk about other options here, the branching enzyme is deficient in which disorder? Branching enzyme. What branching enzyme is doing what in the glycogen metabolism? It is creating the branch. So if the branching enzyme is deficient, the polymer which is formed is going to have less number of branching, and that's called amylopectin. That is called amylopectin. So when amylopectin is deposited, it will result in amylopectinosis, right? It will result in amylopectinosis. So branching enzyme deficiency results in amylopectinosis, which is otherwise called Anderson's disease. Anderson's disease. This is type 4 glycogen storage disorder, where amylopectin kind of biomolecule is deposited in the liver, resulting in hepatomegaly, because amylopectin is going to occupy large space, right? And another option was debranching enzyme, right? So that is again not the answer here. Von Gerke mein nahi hota. Debranching enzyme deficiency. What debranching enzyme is going to do? Debranching enzyme is required in glycogenolysis, which is removing the last residue from the collagen breakdown product. This is the last biomolecule, glucose, which is to be released by cutting this 1,6 bond. So that is done by debranching enzyme. So when that enzyme is deficient, it will result in deposition of this kind of limit dextrin. This is called limit dextrin. The polymer with the lesser number of twigs is limit dextrin. So this limit dextrin kind of biomolecule is deposited, and second, there is some interruption. Just wait for some time. कुछ इंटरप्शन है, आप इसको जरा स्टॉप करिएगा 2 मिनट. अभी क्या करूँ? गड़बड़ हो गया ना, ये ब्लैकआउट हो रहा है. क्या चीज़? ये ब्लैकआउट हो रहा है पूरा लैपटॉप, तो स्टॉप कर दीजिए आप अभी, ऐसे वो तो नहीं पाएगा, अब देखते हैं क्या करना है। ऐसा क्यों हो गया? ऐसे होता तो नहीं है कभी।